Okay, so welcome back from your break, whether you walked away for a little while or whether you just clicked to follow on. Here's our concept question. This is a pretty straightforward one. It's really simple. You decide to breed two purebred dogs. What are the correct genetic terms for your dogs and the subsequent generations? Well, of course, the purebred dogs that you're starting with, those are your parental generation. The generation that you get from that would be the first filial generation, or the F1. And if you breed the first filial generation, you get the second filial generation, or second offspring generation, otherwise known as the F2 generation. So really, the names of each generation doesn't change on the basis of what organisms you're working with. Those are common terms that are used irrespective of the type of organisms that you are using in your genetics experiments. So let's take a look at Mendel's first set of experiments, the so-called monohybrid cross that would lead to the law of segregation. So in these experiments, Mendel started out with pure breeding plants that were pure breeding for purple and that were pure breeding for white. He crossed those to get the first filial and noticed that the first generation plants, the first filial generation plants, were all purple. So there's a fundamental question here. Is the purple, is the white lost? Gone, or is it hiding? So this is why Mendel knew he needed to go on and do the F2 generation, because if the white is truly lost, if purple is the only result here, is the only color that is preserved in the F1 generation and subsequent generations, then he knows what to expect. All the offspring are going to be purple. But if the white is simply hiding, then there's the real possibility that the white will re-emerge in the F2 generation. All he needs to do is produce the F2 generation, hence the need to go on and make the F2 generation. So in some cases, he allowed them to self-pollinate, or in other cases, he transferred pollen, the notion of cross-pollination. In each case, the white did re-emerge, but it didn't just randomly re-emerge. Once he counted up all the purple flowers and the white flowers, he noticed that the ratio was very close to 3:1. It wasn't exactly 3:1, but it was reasonably close. Now we have to remember that the flower color was not the only character that he was working with. Remember, he was working with 7 characters in total. So here are the results for all 7 characters that he worked with. So here's the purple to white, not exactly 3:1, but reasonably close. Seed color, yellow versus green, with the same relationship, yellow to green. So yellow was the one that turned up only in the F1s, then yellow and green turn up in the ratio of nearly 3:1, almost exactly 3:1 in the F2 generation. Round versus wrinkled, very close to 3:1. Pod shape, inflated versus constricted, very close to 3:1. Pod color, a little bit further away from 3:1, but in the ballpark. Flower position, pretty close to what the flower color was. So perhaps it's, you could say it's pretty close to 3:1. Stem length, tall versus dwarf, again, in the ballpark of 3:1. What Mendel realized, however, was while they might be in the ballpark of 3:1, that doesn't necessarily that statistically they represent a 3:1 ratio. But here's where Mendel's education came into play. He had a math background as part of his degree. Not only did he pick up some biology, but he also picked up a lot of math, and he had some outstanding math professors at the University of Vienna at the time. And he realized that all he needed to do was take advantage of his statistical background and run a couple of statistical tests to confirm whether these numbers, while not being exactly 3-to-1, with one exception, the seed color, at the statistical level actually represented 3-to-1 as a ratio. So he applied his statistical analysis, and sure enough, while these don't exactly match a 3-to-1 ratio, at the statistical level, they actually do. They really do represent a 3-to-1 ratio. All sets of data represent a 3-to-1 ratio. So if he'd got this once with the purple and the white flowers, that could be just a coincidence. But to get it 7 times with 7 different characteristics and get exactly the same result each time, well, that's not a coincidence anymore. That's something that is a very real result and needs to be explained somehow. And I'm not entirely sure how Mendel went about doing or arriving at the explanations, but I can show you at least one way in which he may have thought about it by looking at the next diagram. So one of the first things to realize is that one set of the offspring here provide the key to understanding everything. And it's these purple flowers in the F1 generation. And the reason why they represent the key is because they are the product of 2 very different parents. And as a consequence, they must have received 2 very different sets of genetic information. Remember, the purple flowers are purple because purple is fixed, they're pure breeding. So the only possibility that they could pass on to their offspring had to be genetic information, we would say the allele, for purple flowers. On the other hand, the white comes from white pure breeding plants that only can provide information for white. So it turns out, and Mendel realized this pretty quickly, that each individual must have 2 separate pieces of genetic information, we would say 2 separate alleles, for each trait of the character, for each character that they show. So this is one conclusion that Mendel reached based on this observation. And he reached this conclusion again by examining not just the flower color, but looking at all of them and seeing the same result. Basically, you see the same thing. Now, he also recognized that in the F1s, it was only one of the traits that turned up. The other trait did not appear in the F1. And so he coined the term dominant and recessive. Bring it back. So the idea of dominant versus recessive, this was another idea that Mendel proposed, and his argument was that his definition here is that the dominant trait is the one that appears uniquely in the F1 generation, and the recessive trait then reappears in the F2 generation, and it's the one that is less frequent in the F2 generation. Now, in order to try and explain the 3-to-1 ratio, I'm not sure how Mendel went about doing it, but in the modern day, we use what is referred to as a Punnett square. And here, I have a problem with the Khan Academy video that I've got posted, because there it is proposed that the term punnet was applied to the square because the punnets for strawberries at the supermarket are square, and hence the name Punnett square. The reality is that there really was an English biologist named Reginald Punnett. He was one of the 3 individuals who in 1901 separately recognized the significance of Mendel's work. And he developed the Punnett square to allow you to be able to predict the likely offspring of any particular cross by being able to identify the gametes that each of the parental plants are, or parental organisms are producing, and what those combinations of gametes will yield in the offspring. So Punnett square is a really good tool for doing that. Mendel didn't have those, or didn't have the Punnett square, so I'm not entirely sure how he figured this out, but nonetheless, he was able to figure out exactly how this happened. Because we know that in meiosis, one of the things that happens is that homologous pairs of chromosomes get separated. And when we draw genetic combinations, so when we write big P, or we write big P, or we write little p, what these actually represent they represent a pair of homologous chromosomes. And whatever alleles happen to be on that pair of homologous chromosomes. So when we're talking about flower color, there's a single gene for flower color. It's located on a pair of homologous chromosomes. The pure breeding purple flowers have the purple allele on both homologous chromosomes. The white flowers have the white allele on both homologous chromosomes. The parents obviously pass on one of the 2 homologous chromosomes, and that establishes a new pair of homologous chromosomes in the purple flowers of the F1. But because they get a homologous chromosome with the purple allele from their purple parent and a homologous chromosome with a white allele from their white parent, what they have now is not an identical pair of alleles. They have 2 different alleles. And we have terminology to describe those, which I'll mention on the next slide, because we'll show it on the next slide. But we know that when these undergo sexual reproduction, they must undergo meiosis to produce the gametes, the eggs and the sperm, or the eggs and the pollen. And so these alleles on their homologous chromosomes are going to get separated or segregated. And we know that that happens when? In anaphase 1 of meiosis 1, when one member of each pair moves to opposite ends of the spindle. That's when the homologous chromosomes get separated. And of course, that also separates the 2 alleles that are sitting on that homologous pair. When fertilization occurs, we reunite the chromosomes to reestablish the homologous pairs, but it then depends on what are the alleles on each of the chromosomes that are being received from this parent, because there are 2 alternatives. They could receive a chromosome with a big P, or they could receive a chromosome with a little p. Now, just a comment on the big P, little p situation here. In Mendelian genetics, and only in Mendelian genetics, the letter that designates the alleles is chosen on the basis of which of the traits is the dominant trait. So in this case, it's the purple flower color that's the dominant trait. So we choose P as the letter to designate the two alleles here. The uppercase P is used for the allele for the dominant trait, and the lowercase p is used for the allele for the recessive trait. And this is a common thing that we do in Mendelian genetics, and only in Mendelian genetics and genetics that is related to Mendel's observations. You'll see a different way of representing alleles and genes when we do chapter 15 in the next presentation. So here is the gametes from this parent. There's either going to be a big P gamete or a little p gamete. Here are the gametes from the other parent, either a big P or a little p. Then we get the four possible combinations: two big Ps, a big P, little p, another big P, little p, or two little p's. Now, of course, anything that has two big P's is going to be purple. Anything that has the big P, little p, because purple is dominant, is going to be purple. Anything that is little p is going to show the recessive trait, the white. So this was the other thing that Mendel realized. In order to show the recessive trait, the individual had to have two identical alleles for the recessive trait. Any other combination that included at least one of the big P alleles, or the allele for the dominant trait, meant the individual showed the dominant trait, which again is part of the definition of the dominant trait. You only need one copy of the allele for the dominant trait to actually have the dominant trait, at least in Mendelian genetics. As you'll see, it's not always the case. So this explains the 3-to-1 ratio, because these three are going to be all purple. This one is going to be our white. And you can make the same explanation for all 7 characters, because remember, they all behave exactly the same way. So one of the things that we now need to recognize is that we need to differentiate between individuals that have the same alleles on the homologous pair versus individuals that have two different alleles on the homologous pair. And I did jump over a diagram, which I think I need to go back to for a moment, because it just reinforces the idea that we're dealing with homologous chromosomes. Maybe I ought to move this picture to another location a little bit later in the presentation, but it's here for now. So if we look at the two homologous chromosomes at the locus, remember that's the physical position of the gene on the chromosome, you see two different alleles. So here is the locus for the flower color gene. On one of the chromosomes, it's the purple allele. On the other chromosome, it's the white allele. And so these individuals will be purple. Now, in the case of purple and white flower colors here, we actually know what these alleles are coding for. So the purple allele codes for the production of a purple pigment. The white allele actually codes for the production of no pigment. So it's an allele for lack of pigment, you could say. So if you have two alleles that say make no pigment, then you're going to be white in color. If, however, you have two alleles for purple, you're obviously going to be purple. If you have the purple allele and the white allele combination, then the purple allele says make purple pigment. The white allele says make no pigment. So the only pigment you have present is purple. So obviously you're going to be purple. And that's why purple is the dominant trait. Because if the white allele says make no pigment at all, and the purple allele says make purple pigment, then the pigment that's going to be present is purple, and so you're going to be purple. You'll see that in snapdragons, that's a little bit different. So the two terms that I was talking about, if the alleles are the same, remember the prefix for the same is homo. So we say that if the two alleles are the same, they are homozygous. The prefix for different is hetero, so individuals that have two different alleles are said to be heterozygous. So a homozygous could be two purples or two whites. A heterozygous would be a purple and a white. Now that in turn poses a problem because you've got two different ways to be homozygous. You've got homozygous for purple and you'll notice that I put a bar across the tail of the P to indicate that it's lowercase. Most geneticists and biologists do this if there's a possibility of confusion over which letter you're looking at. So if you see a bar over a letter or a bar under a letter, it's just designating that that's the lowercase letter. So to differentiate these, we say that this is homozygous dominant. And this one, obviously, is homozygous recessive. So that's how we differentiate between the two different types of homozygous. No need to differentiate between heterozygous because there's only one heterozygous, big P, little p in this case. Then the other two terms that you need to become familiar with are the terms phenotype and genotype. So when we're talking about the actual alleles that are present in the organism, we're talking about its genotype. When we talk about phenotype, we're talking about what it looks like. So those are two simple ways of understanding the difference between phenotype and genotype. Now, why talk about those? Because when we come back and revisit the F2 generation here, what we recognize is that the 3-to-1 ratio that we've been talking about, well, that refers to the phenotype. So the phenotype ratio is 3-to-1. But the genotype ratio is anything but. The genotype ratio is actually 1 to 2-to-1. So you'll notice I put in the dominant and recessive to the two homozygous so that we can see the differentiation. So there's one homozygous dominant, two heterozygous, one homozygous recessive, which means the genotype ratio is 1 to 2-to-1. And these are absolutes when it comes to Mendel's monohybrid crosses. So if you're dealing with a character whose traits are inherited in a Mendelian fashion, in other words, it follows Mendel's law of segregation, then in the F2 generation, the phenotype ratio is always going to be 3-to-1. That's an absolute. And the genotype ratio is always going to be 1 to 2-to-1. So if you're given data in a genetics problem, say, for an exam or practice questions, if the data is for phenotypes and you can see that the ratio is close to 3-to-1, then you can be confident that you're dealing with a Mendelian monohybrid cross. Likewise, if you're given genotype ratio and the numbers come out very close to 1 to 2-to-1, again, you can be very confident you're dealing with a Mendelian monohybrid cross. These are absolute values. And so these are numbers that you can take home and memorize. You can put these in the bank, so to speak. Now, one of the real problems that comes out of this simple observation of genotypes versus phenotypes is that if you are given an organism that shows the dominant phenotype, you have no clue what its genotype is. And remember, one of the principles that Mendel established in genetics problems is always know the genotype of whatever you're starting with. So if you're given an individual who shows the dominant phenotype, but you have no clue what its genotype is, you can't do anything with it. It would be nice if there was a very simple and quick way to figure out what the genotype is. And fortunately there is, and it grows directly out of understanding what Mendel's monohybrid cross is telling us. This is called the test cross. We still do test crosses today. And as you'll see in chapter 15, the test cross, or the failure of a test cross to give the result that was expected, led to a whole new understanding of the behavior of chromosomes during meiosis. But more of that when we do the chapter 15 presentation next time. So obviously test crosses are really important to understand. So how can we go about doing this in such a way that we can make sense of what the unknown phenotype is in terms of its genotype? Well, it all comes down to making sure that we do the cross against the one phenotype that we know for certain its genotype, and that is the recessive. Because remember, in order to show the recessive phenotype, you have to be homozygous for it. So you know that those individuals are absolutely little p, and therefore they can only make one contribution to the offspring, a little p. So any difference that occurs in the offspring can't be because of what the white parent is bringing to the table, because they can only contribute a little p. It must be whatever the purple parent is contributing that must be making the difference. So given that the purple flower could either be homozygous or heterozygous, it must be the product of these two that must create the difference. So if the unknown is big P, which is this Punnett square, then this parent is only going to provide the little p, which you can see here. That's the only contribution they can make. This individual, of course, is only going to contribute a big P if they're homozygous for it. So all the offspring are going to be big P, little p. They're going to be heterozygous, which of course means they're going to all be purple. On the other hand, if this individual is heterozygous, pretty ugly arrowhead, let's clear that up. This parent is still only going to be providing little p's. But now there are alternative outcomes from the purple parent. They can provide the big P or they can provide the little p, which is going to give us two types of offspring. There will be offspring that are going to be heterozygous if they get the big P from the purple parent, but there are going to be offspring that are going to be homozygous recessive and therefore be white if they get the little p from both parents. And so if the unknown is heterozygous, then you're going to get half purple, half white offspring. Versus if the individual is homozygous dominant, then all of the offspring are purple. Very easy to pick the difference. Either the offspring are all purple or they're half purple, half white. No confusion there. And so by doing a test cross, you can very quickly figure out what your unknown individual is in terms of phenotype versus genotype. And remember, the phenotype is the one thing that creates the confusion, because there are two different ways of showing the purple phenotype, two different genotypes that lead to that. Whereas for the recessive phenotype, there is only one genotype that leads to that. So let's take another break. Here are the concept questions that I'll answer for you on the other side of the break. So if two organisms have the same genotype, must they have the same phenotype? If they have the same phenotype, must they have the same genotype? Interesting questions. Mendel showed that all organisms have two alleles for every trait. Where do these alleles come from? I'll see you on the other side of the break with the answers to the questions.